Let's begin with transcription. This is the first stage of gene expression, and during this process, a DNA template is used to produce a complementary strand of messenger RNA, also known as your mRNA. The base pairing rules are straightforward. A pairs with U, the T pairs with A, the C pairs with G, and the G pairs with C. This gives us a messenger RNA sequence that can later be translated by the ribosome. The second stage of gene expression is known as translation, and it occurs in the ribosome, where the mRNA strand is read 3 nucleotides at a time. These 3 base units are called codons, and each codon corresponds to a specific amino acid. Using the codon table, we can determine exactly which amino acids are produced. So, for example, the AUG codes for methionine, which is the universal start codon, while the UAA, UAG, and UGA are stop codons, which signal the end of translation. Now let's apply these principles to real transcription and translation exercises. We'll move through each example by identifying the DNA strand, producing the mRNA through transcription, dividing the mRNA into codons, and then translating each codon into its amino acid. Let's start with example 1. The DNA strand, which starts with the letter A and ends with the letter C. Using the transcription rules, we convert it into the mRNA strand, which starts with the complementary letter U and ends with the complementary letter G. Next, we divide this mRNA sequence into codons, and then using our codon table, the amino acids coded, which usually begins with the universal start codon, are the methionine, you have the leucine, asparagine, valine, phenylalanine, and finally a stop codon. In example 2, the DNA strand, which begins with the letter C and ends with the letter T, is transcribed into the mRNA sequence, which begins with the letter G and ends with the letter A. When divided into codons, using the codon table, we translate them into methionine, leucine, asparagine, glycine, and the stop codon, which represents the UGA. Here we have a third example with the DNA strand starting with letter T and ending with letter T. After transcription takes place, we then have an mRNA strand or sequence beginning with the letter A and ending with the letter A. When we translate the codons using the codon table, we get methionine, proline, and then a stop codon, which ends translation early in this example. To summarize, transcription and translation follow a very structured and predictable set of rules. The DNA produces mRNA through complementary base pairing. The mRNA is divided into codons, each consisting of 3 nucleotides. And using the genetic code, each codon corresponds to an amino acid or a stop signal. By applying these steps carefully, you can translate any DNA sequence into a chain of amino acids.